Hey everyone, welcome back for more biotechnology. So we are going to continue our discussion on proteins by discussing a very important kind of protein in this particular video, and that protein is the enzyme. So enzymes are biological catalysts. So a catalyst is going to be any material that speeds a process up. So in the case of biology, a catalyst is going to be some material that speeds up the rate of a chemical reaction. It allows that reaction to happen much more quickly than it would otherwise. So an enzyme, when we say a biological catalyst, an enzyme is something that is actually produced by cells. So enzymes in most cases, and we'll kind of point out the exceptions to this, in most cases enzymes are going to be proteins. So what cells can do, and this is really powerful and really slick, cells can produce enzymes because we know how proteins are made at this point. Cells can produce enzymes for the purpose of speeding up certain processes inside the cell. And the nice thing about an enzyme and a catalyst in general is that by definition, a catalyst is not used up in the course of the reaction, nor is it changed by the reaction. So the enzyme in this case, because it is unchanged and because it is unaffected itself by the reaction, it can do the reaction again and again and again. It can oversee and catalyze many reactions again and again and again. The only limit is the stability of the protein. Eventually the protein's going to kind of wear out and need to be destroyed inside the cell, but otherwise there should not be a limit to the number of times an enzyme can catalyze a reaction. So like I said, almost every enzyme is going to be a protein, almost every single one, but there are a couple of exceptions. The most notable exception that we've already seen is the ribosome. So the ribosome had an enzymatic activity called peptidyltransferase. It's what allowed the ribosome to form peptide bonds between amino acids that are attached to tRNA molecules. Well, so the ribosome is a amalgam of both protein and ribosomal RNA, but the enzymatic activity of the ribosome actually does not belong to the protein part of the ribosome. It actually belongs to the ribosomal RNA portion. So in this case, this is what we call a ribozyme, an RNA molecule that actually has enzymatic activity. But otherwise, almost every enzyme is going to be a protein. So enzymes are going to definitely be of interest in the biotech industry, and most of the enzymes that we would be interested in are either going to be the type that build or form chemical bonds between molecules, or those that break them down. Just to give you a couple of examples, the enzyme Taq polymerase is an enzyme that is taken out of a thermophilic bacterium called Thermus aquaticus. That's why it's called Taq polymerase, Taq for Thermus aquaticus. So this enzyme is a type of enzyme that builds DNA molecules out of a template. So once we start talking about polymerase chain reaction in later chapters, this is the enzyme that is necessary in order to make that possible. We've already talked about restriction enzymes back in chapter 4. These are what are called nucleases, enzymes that break the phosphodiester bonds that attach nucleotides together in DNA and RNA molecules. So we can use restriction enzymes to break DNA down and produce smaller fragments from a bigger piece of DNA or RNA. Okay, so let's talk turkey as it pertains to enzymes themselves. So enzymes have particular molecules that they like to work with. So these are called substrates. So the molecule upon which an enzyme works and catalyzes, that is called your substrate. Most enzymes are very specific for one particular type of substrate. So most enzymes are not promiscuous in any particular way. So most enzymes are specific for one type of substrate because the enzyme has what is called an active site that has a very unique shape to it. A unique shape that is such that only one particular type of substrate is going to fit into that active site. You can see this illustrated down here in the bottom right. The enzyme is in the baby blue color, and you can see the surface of the active site highlighted in that red color. So you'll notice that the substrate can fit into this active site because the shape of the active site is congruent with the shape of the substrate. The substrate and the active site both go together well here. A different type of substrate that doesn't have that kind of shape probably wouldn't fit into the active site, and the enzyme wouldn't be able to do anything with that particular substrate. The active site is lined with particular amino acid side chains that are going to participate in the catalysis of the reaction. So a lot of times you'll see enzymes that have acidic or basic side chains in the active site, and those side chains can either accept electrons or donate electrons to the substrate in order to facilitate the breaking of or forming of chemical bonds or even something else. Once the catalysis is over, what you are going to end up with are called your products. So the substrate is transformed into the products by way of the enzyme catalyzed reaction. Once you have formed your product or products, they will be released from the enzyme, and now the enzyme is free to bind to another substrate molecule and start the whole process over again. So most enzymes can do this process many times in a given amount of time. A lot of enzymes have a rate of thousands or even millions of reactions per second. So these reactions can actually happen at a staggering speed. Sometimes enzymes require more than just their substrate to function. So some enzymes require the presence of what's called either a cofactor or a coenzyme in order to actually stimulate catalysis. Sometimes just having your substrate is not enough. So, for example, vitamin C is a cofactor for some of the enzymes that help in collagen synthesis. Calcium ions are cofactors for the enzyme amylase, which helps to break down carbohydrates. Magnesium ions are cofactors needed for the DNA polymerase enzyme that we mentioned before. So the idea here is that a cofactor will bind to the enzyme and modify and kind of straighten out the active site so that the substrate can actually bind, whereas without the cofactor present, the active site has a slightly different shape that actually cannot accommodate the substrates. So when you get to the point in the biotech industry when you are actually working with enzymes, it's going to be very important to be aware of when and if the enzyme that you're working with requires cofactors, because it would be a crying shame to set up a reaction, you add your enzyme to the reaction, and then once you're ready to analyze how the reaction goes, you see that no reaction happened because you forgot to add a cofactor. Enzymes also have other sorts of requirements that really have nothing to do with the substrates or chemicals that we can add. So let's talk a little bit about the effect of temperature on enzymes. The general idea of just chemical reactions is this. Chemical reactions tend to speed up as the temperature rises. The idea there is that as the temperature rises, molecules start moving around a lot more quickly, and the chances of particular reactants colliding with each other increases. So, generally speaking, increasing the temperature is a good thing to do if you want the rate of reaction to increase. But this is not necessarily the case for enzyme-catalyzed reactions. Enzymes, as it would have it, actually require a very narrow temperature range in order for them to efficiently function. So what you're seeing here is a graph that maps out the optimum temperature for a particular enzyme. So you'll notice that the peak of this graph, meaning the temperature at which the enzyme catalyzed reaction is at its maximum, is about 37 degrees Celsius. Any cooler than that, and you'll notice that the rate of the reaction tails off quite a bit. The reaction slows down a lot. So that actually makes sense, and that would be true for any other type of reaction, enzyme or not. If the temperature is too low, the substrates are going to be moving around too slowly to really ever encounter the enzyme at a significant enough rate. So when things get colder, reactions slow down. That's true no matter what. On the other hand, at higher temperatures, it is true that substrates are going to be moving around a lot more quickly, but the problem here is not the substrates, it's actually the enzyme. At higher temperatures, the protein structure of the enzyme is going to start to fall apart, and we call this process denaturation. Basically, We've already talked about how important the three-dimensional shape of a protein is to its function. So if you denature a protein and you force it to lose its three-dimensional shape, guess what it's not going to do anymore? It's not going to function. So once you get the temperature cranked up high enough, most enzymes are going to completely stop functioning, and even though the substrate molecules are moving around faster, without an enzyme to catalyze the reaction, that reaction is not really going to be happening very much. So the graph that you are seeing here that maps out an optimum temperature of 37 degrees Celsius, this would be true for most human proteins and a lot of bacterial proteins. Basically, any organism that maintains either an internal temperature of 37 degrees Celsius, or in the case of a bacterium that likes to live at 37 degrees Celsius, that's generally going to be the optimum temperature for any of those enzymes. So our internal body temperature as humans is 37 degrees Celsius, so all of our cells and all of the enzymes that they make are always operating at a temperature of 37 degrees Celsius, which allows them to maximize the rate of their reactions that they catalyze, and it makes all of our physiological processes go a lot more smoothly. Just as another example, the budding yeast called Saccharomyces cerevisiae, which you know as baker's yeast, it has an optimum temperature of 30 degrees Celsius, which is quite a bit cooler than what our enzymes are accustomed to. So it's the same sort of thing. Yeast enzymes probably prefer a temperature closer to 30 degrees Celsius. If you forced a yeast enzyme to try to operate at 37 degrees Celsius, which is our optimum, it's probably going to get denatured. So basically, your takeaway should be that as far as enzymes and proteins in general go, denaturation is not a good thing. So if you denature a protein, it's pretty much permanently done for. It's not going to really work much anymore. So a lot of times in the laboratory setting, when you're working with proteins or enzymes, you generally want to work at very cold temperatures. So whether you're storing enzymes or purifying enzymes out of cells, you want to be working at a very low temperature to minimize the risk of any denaturation happening. So in these pictures, you're seeing what's called a cold room, which is a laboratory room that is maintained at a temperature of about 4 degrees Celsius, which is generally what refrigeration temperatures are. So you can actually walk into this cold room. You see the door on the left, and then you see kind of what it looks like on the inside. So you can see a lot of buffers and a lot of chemicals and a lot of apparatus that are necessary for the purification of proteins. In addition to having a temperature optimum, you'll recall that proteins have also a pH optimum. We talked about this back in Chapter 3 when we were talking about the pH of buffers. So proteins need to be stored in an appropriate buffer to keep the pH from changing too much. So for this reason, just like what we saw with temperature, proteins are going to have their highest activity within a narrow pH range. In this graph that you are seeing here, you see the pH optimum of this protein around 7.2. Well, that makes sense because 7.2 to 7.4 is approximately the pH of human blood plasma. And we have a lot of enzymes that circulate throughout the blood, so it's definitely good to have those guys circulating at a pH that is most suited for their activity. So the idea here is that if an enzyme is exposed to a solution that has a pH that is too far away from its optimum, the protein may either denature if it's a really big difference, and we already know how bad denaturation is, or alternatively, the enzyme may lose some catalytic activity because you're disrupting side chains in the active site, especially those acidic and basic side chains. Alright, so that is going to do it for this video on enzymes. We have one more video in this chapter, and we are going to start talking about gel electrophoresis of proteins, the same way we talked about gel electrophoresis of nucleic acids in the previous chapter. So thanks for joining us, and I hope to see you next time.